Good morning, everybody. Welcome to our very first lecture together, our very first last lecture together. So what we're going to do today is talk about applications of immunology. There's tons of them and that we won't have time to get to, so we're going to just touch on a few of them. How the field of immunology, how understanding how our adaptive immune system works has helped us in not just in sort of understanding how our bodies fight off disease, but how can we create better medicines for all kinds of things, and even how can we understand things that aren't immune related using the tools of immunology. So that'll sort of be the first half, and then the second half we're going to do a review for the next exam. So I asked you to bring your questions, to have thought about what you want to cover. This is going to be entirely student-led. That means that you're going to ask questions and I will respond to them. So it's just going to be up to what you guys are feeling is the most important for you to review. Okay. So one thing we touched on last time was the types of adaptive immunity. And so let's just see if we remember some of these things. What type of immunity protects newborns? Given that they don't have their own mature adaptive immune response. And what type of immunity is this? Write into the chat what comes to mind when you read those questions. What protects newborns that lack their own mature adaptive immune response, and what type of immunity is this? Okay, so in the chat, some good responses. The innate immune response, of course, kicks in right away. Also, some of you have said IgGs, you're right. And where did those IgGs come from? IgGs came from mom, and so that's considered a form of natural passive immunity. Okay. When we vaccinate, what type of immunity are we trying to create? Or what type of immunity do we create? What will be considered the immunity that you get from vaccination? Yes. So what we're trying to do is generate antibodies and memory cells, and that type of immunity is considered active but artificial. So if we compare vaccination to natural exposure, what the difference is, of course, is that vaccines don't expose you to the actual pathogen, whereas a natural exposure would. Both of these are considered active immunity because they both create antibodies and memory cells. But in a natural exposure, you're exposed to the legit, intact, functional pathogen. And in vaccination, you're exposed to something that is either a piece of that pathogen or is a harmless version of that pathogen. Okay. So in the prep, you may remember this story about the nurse in West Africa who contracted Ebola. And what they did to try and save her life was they transfused blood from convalescent donors. What was the hope here, and what type of immunity were they trying to generate? You can answer either of those questions. Like, what type of immunity would be conferred if it was successful, and why would it be successful? Okay, in the chat, I'm seeing a lot of artificial passive, which is the right answer. Good. So artificial, because it's an intervention. Passive, because she's getting antibodies from the convalescent donors, right? And that, of course, is the goal, is to give antibodies, even though she doesn't have time to make her own to save her own life, give those antibodies to her sort of passively from somebody else. So these are some prompts for the review that I want you to do, or that I suggest you do. Let's really quickly have some raise your hands for some of these, like just sort of off the cuff. How would you explain the difference between a primary response and a secondary response? Raise your hand if you want to explain the difference between a primary and secondary response in the adaptive immune system. Brooke, go ahead.
So the primary response occurs after you have like your initial contact with the antigens, and the second response will occur after your second exposure to it.
Great. And then I'm going to ask you a follow-up question, but if you want someone else to answer it, you can just pass. So my follow-up question would be, so if the primary response is your first exposure and the secondary response is what happens on second exposure, what is the physiological difference? Like, how does your body respond differently the second time?
I don't know if this is right, but I'll try. Is it that the secondary response is like memory cells?
Yes, good. And then I see a couple of other hands. I'm just going to check in to see if other people have extra things they want to say. Thank you for that great answer. Stacy, what would you like to add?
In the primary immune response, doesn't your body like produce memory cells like B and T cells? And then in the secondary, it starts to divide the memory cells and something, I can't remember what, something with the IgG, but it's slipping my mind.
Yeah, so in the primary response, there's usually one or two, a very small number of cells that are capable of responding that have the right specificity. And in that primary response, they divide a lot, and some of them are reserved as memory cells. But now those memory cells are a larger population, yes. So in the secondary response, that larger population can come back online. Luke, what did you want to add?
The secondary response is not only a lot faster, but it is overwhelmingly larger.
Yes.
So that's why after we're exposed the first time, the second time we don't nearly feel the symptoms a lot of times.
Yeah. In the secondary response, quite often we don't get sick at all. And that's because we have a faster, more robust response. So actually the antibody titer in our blood is higher because there's a larger number of cells producing antibody and a larger number of cells actually being able to detect the antigen in the first place. So we have faster response and we have a larger antibody response just because there's more cells making antibodies. Okay. Who would like to define herd immunity? Raise your hand if you'd like to define herd immunity. Denise, go ahead.
It's immunity of most of the population.
And do you know what that threshold is, that sort of gold standard?
Is it 90%?
Yeah.
90%?
Yeah, so herd immunity is when enough people in the population are immune that the remaining unimmune population is protected. There's additional protection for them, even though they don't have their own immune response. If there's enough people in the population that kind of will deflect and prevent transmission, that they are protected as well. And 90% is what we aim for, though we can't always get there. Any amount of immunity in the population is helpful, but when you get to that like roughly 90% is when you start to see real gains. Okay, so this is just, you know, you don't have to memorize this. This is not like a test question, but this is just, hey, like this stuff applies to our real lives. We all have experience with this. So what vaccines have you had or that you know about or that have you given to your kids or you've just heard about? Put them into the chat, what things we have as generally available to the public vaccines. And I'll just start reading them off. Okay, we've got the MMR, measles, mumps, rubella. Fun fact, there's some evidence now that there's a little bit of a protective effect against coronavirus if you have MMR. It's obviously not completely protective, or very few people would get sick because a lot of people have had MMR. Flu shot, right? That's the one we got to get every year because right now the flu strains mutate quite rapidly, and there is an effort to create a universal flu based on antigens that don't change, but that has not been produced yet. We've got Tdap, right? The tetanus, diphtheria, pertussis shot, and all of those are childhood diseases that used to be, you know, very severe and deadly, and those are well under control because of that shot. We've got, what else am I seeing here? Hepatitis. All the hepatitises really have vaccines. Hep B is the one that a lot of people are mentioning. HPV, right? HPV. quite a few strains of those, but there's some vaccines that either protect against 2 or 4 of the most common carcinogenic ones. Remembering that viruses can cause cancer. Some viruses do because they're retroviruses. And what else am I seeing? Oh, yes, there is a vaccine against tuberculosis, though not commonly used here. I believe people in the military may receive it, and overseas it's common, which creates complications if you are moving, if you immigrate from another place where that vaccine was used, you're actually going to test positive on the TB test, even though you haven't had TB, so you have to, that's a little bit of a thing. Okay, great. We've got polio, bacterial pneumonia, rotavirus, varicella. Yes, yes. Good. Okay, good job, everybody. Nice work. Okay, so this is a raise your hand question. I want you to think about it for a second. So, well, first let's answer the first part in the chat. Are antibiotics effective against viruses? Just put that yes or no into the chat. Good, okay. So everyone agrees that antibiotics, we're not talking about vaccines right now, antibiotics are not effective against viruses. If you can explain why, raise your hand. And it might take a few answers just to see sort of like, you know, there's a couple ways to answer this. Why are antibiotics ineffective against viruses? Raise your hand. Eveline, go ahead.
Is this because like antibiotics, they attack like the peptidoglycan crosslinks in the bacteria cell wall, and viruses don't have that, so there's nothing for the antibiotics to like penetrate the virus or attack?
Yeah, so some antibiotics target the peptidoglycan, which is a structure in the bacterial cell wall, and so they're antibacterial, and viruses don't have that structure. That's a great answer. Not all antibiotics work that way. So if anyone would like to add to that excellent answer, then raise your hand and sort of give me more. What else would be reasons? You could talk about other specific reasons, or you could talk about a more general reason that antibiotics are not effective against viruses. Stacy, go ahead.
Is it because they're not alive? So you just have to let them run their course. There's nothing really to attack with antibiotics.
So you're on the absolute right track. I'll rephrase it slightly and just say that the antibiotics are specific agents. They target specific things, and it just so happens that the things they target are cellular, and viruses are not cellular. They're not alive. They're not cellular. They don't have cell walls, which is the target of some antibiotics. They don't have ribosomes, which is the target of some other antibiotics. They don't have cell membranes, which is the target of some antibiotics. So all of the specific things that different antibiotics will go after are all cellular structures. And that's why. And the reason for that is that antibiotics are created by microbes to kill other cellular microbes. Now, are there antiviral strategies that cells have developed? Absolutely. We don't call them antibiotics. Those would be different things. Natalia said viruses do not have organelles. Correct. Viruses don't have organelles. Antibiotics don't usually target organelles, although the ones that go after ribosomes can harm mitochondria in eukaryotic microbes. But generally the structures that those antibiotics go after are like, you know, cell wall, cell membrane, ribosomes, that type of thing. Good. Can you vaccinate against bacteria, or can you just vaccinate against viruses? Write that into the chat. That's right. The answer is obviously both, and hopefully that was, you know, at least in the back of your mind because of all the things we just listed. Remember, we just listed all these vaccines. A lot of them are against viruses, right? Polio, measles, but some of them are against bacteria, tetanus, pertussis, diphtheria are all bacterial. Okay, so we've already talked about our 4 types of adaptive immunity, so I'm not going to go over that again. I'm just going to sort of show this slide to remind us that these are the definitions of these things, and these are the, like, sort of also a different way to kind of remember that stuff, right? Naturally acquired active immunity from infection, naturally acquired passive immunity when you get your antibodies from mom, artificially acquired when you get a vaccine, and artificially acquired passive when you get antibodies, injected antibodies. We talked a little bit in epidemiology and about the importance of vaccines and their relative importance, right? They are very effective. In epidemiology, we talked about how they're not the only important factor. If you're going into a place where, you know, you sort of have to start from scratch, then some of the things you want to target that will be very effective would include, besides medical interventions, what kinds of things would you want to make sure people have access to if you're trying to reduce the spread and impact of infectious disease? Yeah, you want to make sure they have access to clean water. Other factors we talked about that weren't medical, proper sanitation. We also want to make sure they have enough food, right? Because nutrition is important to support the immune system. A lot of people can fight off diseases on their own if they have enough of food, and without that, their antibody production is really compromised. Education, political things can impact that. You know, stability of the region. Are you having to flee from a natural or disaster or war or something like that? So vaccines are important, but they are important in context of other things. Once you have like a pretty stable infrastructure, like working sewer system and water treatment and a relatively stable government that you know is not, you know, people are not constantly having to run for their lives, at that point, vaccination is extremely important to bring the caseload down, you know, basically in some cases to zero or low, right? But they, of course, only work if you use them. And so we do see that there's a big impact of these interventions. And then later on, when our caseloads are getting into a more manageable range, vaccines can kind of just take care of the rest. All right. So let me ask you this. This is also straight out of epidemiology. Are diseases like polio, measles, tuberculosis, and whooping cough still a problem, yes or no? Yeah, they're not so much a problem here. I mean, I doubt that there's been somebody in your direct family who had polio or has been exposed to polio, although it's possible, especially if you have people in your family who are recently immigrated from a place where it's still endemic. Or you have long-lived family members who lived through the polio epidemic in this country. Measles has been well controlled until recently. Of course, when people stop vaccinating against measles, it does come back. Tuberculosis has, at least until recently, been well controlled. Of course, now there's a drug-resistant version that's resurfacing, and whooping cough is largely eradicated in most parts of the country, although it's not completely gone. It can come back. But if we look around the world as a whole, these infectious diseases are still a problem. So then if we think about something like coronavirus, why is it having such a major impact? If at least here in the US, barring, you know, reemergent diseases and like, you know, things like measles outbreaks because people aren't vaccinating, or resurgence of STDs that are mutating away from their antibiotics that were previously controlling them, why is coronavirus a problem here? Okay. Yes, good. So we have no immunity. It's brand new. So there's no herd immunity at all. It's brand new, so we haven't had a time to develop a vaccine. And so there is this situation where even in a place where, you know, with public health and infrastructure and medicine, disease is overall well controlled, new diseases can affect people very severely in the beginning. And if they are contagious, then they can spread all over the world and affect all countries. And I think that this country is actually a perfect case study of this. The US is, relatively speaking, a wealthy nation. Not everybody in the country is wealthy, but relatively speaking, there's a lot of infrastructure and access to healthcare and money. And despite that, this country is doing poorly compared to many countries, including third world countries. So when you don't have immunity, when you don't have vaccines, and then when you don't learn the lessons from the past about how to deal with contagious disease, then you can have a very serious epidemic or pandemic, even in a place that would normally not experience such a high level of infectious disease. Okay. Now, one of the things in the application section that you do need to know for the test, and it's pretty cool, is that vaccines are not all the same as each other, right? So there's different types of vaccines. Claudia asked a great question I want to come back to in a second. While I'm thinking about her question, I'd like you to list a type of vaccine that you know of in the chat based on your prep. What were the types of vaccines that we talked about? And then while you're listing those, Claudia asked if there's any research on people who've gotten COVID and if there are antibody tests. And the answer is yes to both those. There's, you know, it's so new that the research is new as well. So there's a lot of people scrambling to do these studies. There's not a lot of time that's elapsed since the first people got sick. So, you know, you've probably heard of people who have what they call long haulers, people who have recovered from COVID, but are still experiencing symptoms that are, you know, sort of longer term. But long haul is a little bit of an interesting term considering it's only been a year. So we don't really know what the long haul is, but there's all sorts of information out there, sort of overwhelming in terms of people who have gotten sick, and some people don't get any symptoms at all, and some people get mild symptoms, and some people get severe symptoms, and some people go on a vent, and some people die, and then some people recover and have long-term symptoms. In terms of antibodies, I'm not entirely sure what Claudia was asking directly, but there's a lot of research trying to figure out if people have long-term immunity, and actually the research is mixed, but it's encouraging. It seems that people do have antibodies against COVID once they get it. And in terms of antibody testing, yes, you can get an antibody test, but I actually would have to go and look and see what the latest is today on which tests are available and what exactly is sort of considered the most effective. So I'll have to come back to that question. Good question. Okay, so let's read these answers. Attenuated whole agent, nucleic acid, subunit, toxoid, and I'm just checking to see if anyone has, there's one missing. So we got attenuated whole agent, we've got toxoid, we've got subunit, we've got nucleic acid. Let's see if the one that I'm looking for, there we go, inactivated whole agent. Good job, you guys. Now here in italics are some examples. You do not have to memorize these at all, but just so you kind of can feel like a connection to them. The MMR, it contains an attenuated whole agent. Okay, that means it's alive, right? It's a virus, so it's not really alive, but it is actually capable of getting into a cell. It just doesn't cause disease. So attenuated means that even though it's still functional and can reproduce, it's not a disease causing anymore. It's been altered through passing through cells over time. One of the polio vaccines that's not commonly used here, but has been used overseas because it's more effective, is Sabin polio version. And then the flu shot is the one you've probably gotten, but there's a nasal version. You can drip it in your nose. That's also not commonly used, but that is an attenuated whole agent. And this one, you can get the flu from it at a very low rate, but this is not the one you probably got. You probably got it injected, which is an activated whole agent. A different version of polio is also an inactivated whole agent. So that's when the whole agent was taken and usually heat killed. So it can't reproduce, it just looks like itself. We've got the subunit vaccines like pertussis. We've got toxoid vaccines like tetanus toxin and diphtheria toxin that were heat inactivated. All right, so I've already said this, but let's see if you were alive and paying attention. What are toxoids made of? Write that into the chat. Be as specific as you can. What are toxoids made of? How would you make one? Good. Yes. And someone had already put that into the chat as a good answer. So yeah, it's a heat-killed version of exotoxin. So whatever the toxins are that bacteria make, those bacteria that make toxins, and when their toxins are what make you sick, those toxins are also antigenic. They're proteins, and they can cause an antibody response. And we take a version of them and we heat kill it so it can't do its job anymore. It can't hurt you, but it still looks like itself. You can generate an antibody against it. All right, how does a subunit vaccine differ from whole agent? How does a subunit vaccine differ from whole agent? Good. That's right, it's just a piece. So let's think about it before we move forward in the next question, what we could make a vaccine out of. Let's start with an easier one. So let's suppose we had a virus. Quite often, people will start by trying to make like an inactivated whole agent out of a virus, or sometimes an attenuated whole agent. But if we didn't want to do that, if we just wanted to make a subunit vaccine, what could we use as one of the pieces? Think about viruses. So we're thinking about viruses right now. What could we use that's a piece of a virus? Okay, so someone said protein. That's on the right track. There we go. So spikes, you could use the protein spike from the virus, or you could use the capsid protein. Now, someone said capsule. Several of you said capsule. Don't get that mixed up. Capsule and capsid are actually different. I know, so annoying that they sound the same. Capsule is bacterial. Capsid is a protein on the surface of the virus, and spikes are proteins on the surface of the virus. Both of them could be antigenic, so that's great. If we were trying to do a bacterial vaccine, almost always it's going to be subunit or toxoid, because like a whole agent bacteria is quite difficult to generate as a vaccine. It's not very, it's logistically difficult and it's more dangerous. So typically we'll take a piece. So think about what are the pieces on the surface of a bacterium. What things are there that we could maybe make vaccines out of? Okay, some membrane protein or membrane carbohydrates, good. Can we think about anything more specific that we know about, like what structure is found on the exterior of some? There we go. We could take a piece of the capsule. Good job, Veronica. Yes, Rebecca said that too. We could take a piece of the capsule, and that's actually commonly done. Or we could take a membrane protein, especially for a bacteria that's not encapsulated. You could take a flagella protein. I actually don't know, Luke, of any examples of that, but in theory, it should work, right? I don't see why not. It may be not the preferred one just because typically the flagella is a smaller component of the bacteria, so maybe harder to see, but I don't know. Okay, so a small piece such as a polysaccharide from a capsule or a spike protein or a capsid protein from a virus. Nucleic acids, how do they differ from other vaccines? Think about this before you answer this question. I think I'm going to make it a raise your hand. I'm going to give you more information for how I want to frame this answer. When we think about all the other types of vaccines here, attenuated whole agent, inactivated whole agent, subunit vaccine, toxoid vaccines, all of these have something in common, which is that the antigen comes into your body and then you make antibodies against that antigen. How does a nucleic acid vaccine differ in terms of that series of events? Some of you are saying it's only made for horses, not humans. And that's true right now. There's only nucleic acid vaccines that are effective in horses, like the West Nile virus vaccine. It's not to say it couldn't be done in people, just we haven't developed one that's effective in people yet. There's scope for it to be effective, but hasn't worked well. Elizabeth, what would you like to say in a response to the prompt?
So instead of like it differs in that when they inject the DNA and then your cells make the antigen and then your body reacts to that antigen and makes the antibodies against it.
Yes, exactly. So let me say that again, because that was a great answer. So basically, all of these vaccines are similar in the sense that whatever they're made of, whether the whole thing or just a piece of the thing or a heat kill toxin, they are the antigen. The antigen is what's injected into you, and you make antibodies against that antigen. But in a nucleic acid vaccine, the nucleic acid is just the instructions for the antigen that comes into your body. Your cells then read the instructions and make the antigen. Now, since it's not a human antigen, right, the thymic selection didn't select didn't kill off cells that would respond to that antigen. When that antigen is detected, your body goes, whoa, what's going on? Let's make some antibodies. But as you can imagine, well, maybe you can't, but I'll help you imagine it. This is both a potentially very powerful, but also potentially like logistically more difficult vaccine to make effective, which is why probably it's only been effective in horses so far. And that's because how do you deliver enough DNA to the right place to make the cells make enough antigen? And on the other hand, if you can make that happen, then you could put the instructions for anything. Like, you don't have to grow it. Sometimes what's holding back a vaccine is that this particular antigen be great, but we have a hard time growing the antigens for some reason. But if all you had to do was inject the instructions, man, like I could make instructions for almost anything tomorrow in a machine, just if I had a way to inject it. So this is potentially very, very versatile and powerful. It could also, like, think about this. If we just had the instructions for the new coronavirus that showed up tomorrow, that's a mutated version of COVID-19, COVID-20, let's say, and all we needed to do is inject a nucleic acid, we just have to be able to read the code, which takes 10 seconds now, and then, ramp up our distribution of the nucleic acid, which is faster than trying to grow up a bunch of actual pathogens. All right, so we move on, since that's not stuff you need to know for the test, just fun. But this is the important point. Your cells, as Elizabeth said, make the antigen in response to these instructions, and then you generate your own antibodies in memory. So you don't get the antigen, you have to make the antigen yourself. Okay, a couple more things. Of the types that we've just described, which one is the most effective? Just answer this first part in the chat. Which is most effective? And then we'll do a raise your hand for the why. Yes, the attenuated whole agent is the most effective, typically. So raise your hand if you can explain why that's the case. What is the deal here? Why is attenuated whole agent, typically speaking, the most effective vaccine? Raise your hand. Serena, go ahead.
I'm gonna take a guess off of what I know about the subunit. So I know for the subunit, it's only one antigen. So for the attenuated, it's multiple, correct?
Yes, good job. That's right. So the whole agent is the whole enchilada. Every antigen that you could possibly respond to is there, which means there are more cells in your body that are probably going to respond, which means your response will be faster and more robust. Claudia, you had something you wanted to add to that?
I don't know if it's this. I don't know if it's this one that I'm thinking about, but doesn't this one also reproduce?
Yes, that's also true. It's able to reproduce. So we get a faster response, basically, and we get a sort of more widespread response. Like, just imagine, we talked about this in adaptive immunity, how you all start off with this kind of giant population of very diverse Bs and Ts. And they're generated like by shuffling a little gene that creates the antibody or the T cell receptor. And there's so much shuffling that we're sort of like primed to respond to anything almost. But you could be the unlucky person who just does not have a B or T that responds to the particular antigen that's chosen for a subunit vaccine. That happens. But if it's the whole agent, that chance is way lower because there's so many antigens. So chances are good that you have at least something that's going to respond to that. Plus there's so many things, right? So many things to respond to. So more cells are going to be creating more memory cells, and it's just a much better response. Okay, good answer. Which of these, sorry, not which of these, given that this is the best one in terms of being effective, what is the risk that's associated with this that you don't get with the other vaccines? What's the risk? Raise your hand. Serena, go ahead.
Isn't it that you risk it mutating and like turning back to like that viral form versus it
Yes,
just staying in the activated like antigen form?
that's right. Because it hasn't been inactivated, it's still capable of reproducing. And when it does so, it can mutate. And the mutations that were originally done in order to make it harmless could be undone. And it could go back to a virulent form. Now, that's extremely rare. It usually is like a 1 in 2 million shot, all right? On an average for these types of vaccines, if you sort of look at typically these attenuated whole agents like measles or one of the polios, it's like 1 in 2 million times that you're going to actually get sick from the vaccine. And so if you're vaccinated against something that is more than 1 in 2 million times likely to get you, then it's a risk worth taking. But if that's not the case, then you might want to reassess your risk. Okay, so here again is the example of, just so we know like what we're really up against. Same with the nasal flu. Remember I told you that the flu shot is inactivated. You're never going to get the flu from it. But the nasal version, which you probably have not had, is attenuated. So there is a low risk, something like this. I don't think it's exactly this, it's something like this, where you could get the flu from that. Okay, and we've already kind of touched on this with some of our previous answers, right? One of the pros of the subunit is that you're never going to back mutate to anything. You can't be harmed by it unless you're allergic to that antigen. And one of the cons is that because it's only one antigen, it's not going to be as effective. Okay. Now, when we're making a vaccine, unless we're making a DNA vaccine, we need to culture whatever that microbe is. And if it's bacteria, we have to culture the bacteria, and then we got to chop it into pieces. If it's viruses, we've got to culture the viruses. So how do we culture viruses? This is straight out of chapter 13. How do we culture viruses? What are our options? Okay, so we can grow it in its living host. For instance, if it's an animal virus, we could grow it in a living animal. If it's a bacteriophage, we could grow it in a culture of bacteria. Animal viruses can also be grown in fertilized eggs, have to be embryonated fertilized eggs, that's right. Or in cell culture, right? So we could take the cells out of the original animal and grow them artificially and grow them there. Okay. So we can grow them in cell culture in the intact host and the embryonated egg. Can we grow them? Like if it's, let's say it's any virus, could you just get enough food together in a broth and put the virus in the same way you'd grow bacteria? Okay, people are saying no. So raise your hand and tell me why. Why doesn't this work? Why can't you just put food together and pop it into some food? Raise your hand. Luke, go ahead.
Because it needs to use the living cell structure as the machinery to grow and reproduce itself, and food does not have that.
That's right. Unlike cells, which just need nutrients, because they have the machinery to use those nutrients, viruses don't. What they have is genetic information. So what they need to do is get into a cell, have the cell make proteins that are going to help the virus do what it needs to do. And then it leverages the ribosomes of the cell and the metabolic abilities of the cell to generate energy, and basically all the cell's machinery. So because viruses are not cellular, they don't have a protein making machine like a ribosome. They don't have metabolic ability, so they can't generate energy to do synthetic activities. Okay, so when we talk about vaccines, when vaccines are developed, one of the things that kind of, slows down their production is we've got to take a minute to evaluate their safety. And there's something called the therapeutic index that kind of evaluates the risk versus the benefit of the vaccine. So where's the risk coming from? Let's say it's an attenuated live virus. Where's the risk coming from? You can put that in the chat, or you can raise your hand. I'll take either one. Mutation, yes. Attenuated live viruses have a risk, even though they themselves are not harmful because they've been mutated to the point where they don't cause disease anymore, they could back mutate. And again, that risk is quite low, but it's not zero. Now, most vaccines can have side effects. Write into the chat what those side effects might be. What are sort of common side effects of having a vaccine? You might experience pain at the injection site. Very common to experience redness and swelling. You might develop a low fever. And if you're unlucky, you might have an allergic response to some of the components of the vaccine. So very common, redness, pain, and swelling. Now, what is the process that's happening here? Why is this happening? What's the reason for the redness, the swelling, and the pain at in the injection site? Yeah, that's the inflammatory response. That's your innate immune response kicking in saying, whoa, something foreign came into the body. And it'll start even before the adaptive immune response has kicked in. And this is rare, but it is something you need to be aware of. If there's a component of the vaccine that you're allergic to, and most common thing would be if it was a vaccine made in an embryonated egg, very hard to purify away the egg proteins. So you could have a severe allergic response to a vaccine that's been created in eggs if you have an egg allergy. And there are some other things that rarely people can be allergic to. So you have to sort of be aware of that. All right. So here are some questions to sort of feel, comfortable answering, not because you necessarily have to memorize these factually for the test, although some of them you do. But we want to understand like what happens when we are vaccinated. So the thing that happens is we do make plasma cells right away that pump out antibodies. So we will have an immune response right away. But we then, most importantly, make T and B memory cells for later. And these are now a much larger number of cells that are patrolling your body for the same antigen that you originally might have had like 1 or 2 cells able to respond to. Now you have like a couple thousand. We need boosters quite often, especially the first time, but throughout life, we need boosters for various things. Like often you've got to re-up your tetanus, and that's because memory cells don't live forever. You are not immune from colds, even if you have a cold, because unfortunately, and this sucks, colds are caused by many strains of viruses in 2 different genuses. And they do have a mutation rate as well. But this is the most important thing. It's just that there isn't just one cold. There's like, 200 versions of the cold. So you've got to catch 200 colds before you're immune, and then you got to start over because probably it's been a while. You need an annual flu shot, right? Because that flu is mutating at a high rate. And remember, that flu is the perfect storm. It's got an RNA genome, so that polymerase that makes it as error prone. It's got multiple pieces and multiple hosts, so they can easily swap. Children and elderly people at highest risk because of their lowered immune response. And with that said, occasionally you have this weird situation like in the 2013 flu epidemic, where the older people were protected and the younger people were like people in their 20s were getting the flu very seriously. And that was because it happened to be a strain that had been around, 50 years ago. And there were people who were already immune. Okay. So now let's talk about applications for reals. The understanding that we now have about how your adaptive immune response works and how antibodies are generated and what they can do has been incredible, not just in the treatment of infectious disease, but actually with far-ranging consequences for medicine and diagnostics. So one of the things that antibodies can do is help us to better diagnose patients. And also, antibodies have been leveraged as cures for various diseases, some of which are not even infectious diseases. Let's first talk about the process of making antibodies, okay? Now, there's lots of different types of antibodies you can make. You can even make camel antibodies and llama antibodies, which I'm not gonna lie are my favorite ones just because it's fun to say llama antibody. But the most common way to make a monoclonal antibody, that is, if you figured out this particular antibody is useful in whatever context it's useful in, and we wanna make a whole bunch of identical ones, this is the way that you typically do it. You take the antigen that you are trying to make antibodies against. You inject it into a mouse. A mouse has an immune system very much like yours. In the spleen is where all the B cells are, so that we take the spleen out, unfortunately, and you may have guessed this already, the mouse does not survive this. You separate all the B cells out, and then you put them through a process of hybridization. You mix them with a line of B cells that are cancerous. And why you won't want to do this is because you're trying to make a Frankenstein version of the cell. These cells are B cells, but they live forever because they're cancerous, and they've forgotten how to make their own antibodies, so they don't have their own antibodies. So we mix them together, we make a hybrid cell. We select only for the hybrids, and now each cell has the ability to make one and only one antibody, because of course, remember, this mouse is just like you. When this antigen goes in, there's lots of different cells in the body that could respond to it, and it's not just going to be one antibody that's created by this antigen. It's going to be several different ones, and we don't know which one of those ones we want necessarily. So we want to take each individual cell, because of course each cell makes only one type of antibody. We separate them all out, we grow them up. Now that we're amplifying a clone of them, everybody here is the same, and they make one type of antibody, and everybody here is the same, make a different kind. And we test each one until we find the one that we like the best and does whatever we want it to do. And then we just grow that up forever, because remember, these are immortal cells. So this is incredibly powerful and effective technology. Okay, and just to give you an example, and you're gonna go look this up yourself in a second, here are just a few examples of monoclonal antibodies that are used not in the treatment of infectious disease, but actually in the treatment of cancer and autoimmune disorders and other diseases that you might not have thought of as being something that we could treat with antibodies, and now we can. So I haven't told you what exactly these do. I'm gonna give you a second to pick one of these and go look up what you can find out about it on the internet. Really what you're trying to figure out is, what specifically does this antibody do in this context? Like, can we figure out why it's effective for helping people with kidney transplants, or for helping to treat Crohn's disease or leukemia or breast cancer? What is the target of this antibody? See what you can find out. I'm just going to give you a few minutes to go look that up. And then I'll be back in just a couple minutes, and I'm going to see what you came up with. Great. So I just want you to write into the chat whatever one of these you chose next to whatever information you found, and I'll just see what you came up with. Good. So April said infliximab, it disrupts the pro-inflammation cascade. And Amanda said rituximab is a monoclonal antibody against the protein CD20 found on the surface of immune B cells, and it triggers cell death. Shannon says miromanab CD3 is a monoclonal antibody that reduces the body's natural immunity for transplants. Stacy adds about infliximab that it binds to TNF alpha, helping to prevent inflammation. That's right. Cambria says rituximab binds CD20 on B cells. Good, good. Yes, miromanab reduces your immune system so the white blood cells won't reject the kidney transplant. All right, lots of good answers in the chat. You guys did a fabulous job. My goodness, wonderful. Okay, so yes, these are all targeting specific things on different cells in your body that are involved in some kind of immune response. So this miromanab targets a protein on T cells, and it reduces your body's response to the kidney transplant. So it helps you not to reject your kidney. Infliximab targets tumor necrosis factor, and this is a cytokine that's involved in inflammation. Okay, now Crohn's disease, I don't know how much you know about it, but basically it is sort of considered an autoimmune disorder. The etiology and the causes and the progression are all sort of still under a lot of study, but it causes lesions throughout the colon and the small intestine that can eventually be so severe that you actually need part of your or all of your large intestine removed, and it can cause a lot of pain, reduce your ability to absorb food, and so this is an inflammatory disease, basically. Not entirely clear why people get it, but there's genetic factors, environmental factors, and mysterious factors. But by reducing inflammation, you can reduce the severity of this disease. Whoops, sorry about that. This is a drug that does a lot of things for different conditions, but includes one of the forms of leukemia. And so it targets a protein on the surface of B cells. And that is a B cell that's part of the leukemia. We've got this here, which is one of my favorite ones. This is one of the very, very first monoclonal antibodies, and some of you may actually have heard of this. Its trade name is Herceptin, and it was a miracle drug against breast cancer. Not all breast cancers can be treated with this, but there is a protein on cancerous breast cells, some of them called HER2. Well, all breast cells have it, but there's some of them have either too much or an overactive one that causes the cells to divide too much. And if you have that particular form of breast cancer, this is an incredibly specific and effective treatment. It was completely revolutionary when they discovered it. Okay, good job, you guys. So, in addition to this sort of ability to use antibodies as very specific drugs, remember, think back to that sort of search for the magic bullet. If we want to think about, like, trying to develop a treatment that doesn't give you as bad symptoms as the disease itself, right? Remember when you have an option to treat some STDs with, like, poisons, basically. So these antibodies are so specific. They target very specific things, and that makes them even better than antibiotics in treating infectious disease, or in treating various kinds of disease, including things like cancer that you wouldn't think of normally as being treatable with antibodies. But they're also really valuable because of their specificity in diagnosis. So when we develop an antibody for a diagnostic test, there's a couple things we have to worry about. One is sensitivity. Sensitivity is a measure of how likely that the antibody will respond. So basically, if someone is positive for an antigen, is the test sensitive enough to detect that? Like if they just had a really low level, let's say it was very early in HIV infection and the actual viral titer was really low, would this test be sensitive enough to pick it up? So sensitivity, we want to be sensitive. We want it to be able to pick it up, to be able to bind that antigen really well. And then we have to worry about specificity because some antibodies bind to more than one antigen if the antigens look very similar to each other. So specificity means what is the likelihood that if we get a positive test result, it's real? Or another way to say that is, if it's not specific enough, we could get false positives. We could get someone who is actually not infected with whatever we're looking for, or doesn't have the drug that we're testing them for, but actually the test says they do. One of the earliest types of diagnostic tests that rely on this idea of leveraging the immune response to detect things is this experiment that Koch did actually with tuberculosis. He had guinea pigs that already had tuberculosis, and then he injected them with a little sample of it, and the site of injection became red and swollen because, of course, what was happening is they already had antibodies against tuberculosis, so when the new injection showed up, the site reacted. You saw inflammation at the site. So does this remind you of anything? Does this remind you of any test that you've had? Serena, go ahead. Excellent, yes. And other people in the chat have also mentioned this, allergy testing. Now, some of the other people in the chat have mentioned the TB test. So those are two great examples. The allergy test, which I've also had, is where they take a little bit of antigen to find out if you're allergic to it, and they scratch it into your skin and see if it welts. And the tuberculosis skin test is similar, right? When you go into healthcare, they'll test you to see if you've been exposed to tuberculosis. If you have, you should have antibodies, so they put a little antigen on your skin and see if it swells up. I've had that test too when I went to work in a hospital briefly. And of course, if you have been vaccinated against tuberculosis, even though you never actually had the bacteria, you're also going to react on this test. So it's important if you're taking this test and you're, from another country where that vaccine was administered, to say, hey, I've already been vaccinated, so this test is going to test positive. Okay, last but not least, we've got ELISAs, and ELISAs are so cool. It's a word that stands for enzyme-linked immunosorbent assay, which is very long and complicated, but what it means is if you have an antibody against something, you can use that to make a pretty simple color test. And what you do is you've got an antibody linked to an enzyme, and the enzyme is usually peroxidase. And peroxidase takes a colorless substrate, and when it cuts it, it produces a color. So there's a couple of terminology things that we're going to just sort of see if we can review before we look at a picture of this assay and talk our way through it. But what is a primary antibody in the ELISA? What is a primary antibody? If you know, raise your hand or write it into the chat. What does a primary antibody do, and what qualifies it as a primary antibody? And if you want to look it up, you go ahead and look it up. And if you're not sure, you can put a question mark into the chat. If I see enough question marks, meaning that everyone's still alive and willing to say, I don't know, we can move on. There we go. Thank you. Okay. Genesis raised her hand. Go ahead. Yes, that's correct. So, and this suggests there's more than one antibody, which is true. There's a secondary. So there's a couple of antibodies involved in this test, and the primary one is the one that binds the antigen. Okay, so that's right. It binds a specific antigen in this particular test. There's also a secondary antibody. If you know or can guess what that secondary antibody is, you can write it into the chat or raise your hand or put in a question mark. Those are your options. What is a secondary antibody? Even if you just want to wildly speculate. Genesis, go ahead. Yes, that's right. So the primary binds the antigen, the secondary binds the primary, and it's the secondary that's linked to the enzyme. Okay, so the enzyme is important because it's linked to the secondary antibody. So only if the antigen is present, which means the primary bound it and then the secondary bound it, then you get the enzyme, and the enzyme is there so that when you add the substrate, it cuts the substrate and creates a color. But if there's no antigen, then that doesn't happen. We don't see a color change. Okay, so here is an example. There's many ways to do the ELISA, but this is one example. This is commonly done in diagnostics where we're looking to see if somebody has an antigen. For example, do they have a particular viral infection? Do they actually have viral particles in their body? Okay, so we would put their sample into a plastic well that binds to that antigen. Okay, so now this is the patient sample. If they don't have the antigen, whatever it is, let's say it's a virus, then this step won't happen, right? It'll just stay clear. The next thing is we add the primary antibody. Remember, what does the primary antibody do, guys? Write that into the chat. What does the primary antibody do? It binds the antigen. Yes, good job. It binds the antigen. So there it is, binding the antigen. Then, what's the next step? After the primary, what do we add next? We add the secondary. Good. And what's special about the secondary? What's this doodad here? What's on the secondary? It's the enzyme. Okay, so the secondary binds to the primary and it brings the enzyme. And then we add the substrate. And if all of this happened, then we get a color. But if there was no antigen, then this wouldn't happen and that wouldn't happen and this wouldn't happen. So we either get a color or we don't get a color depending on whether you have, let's say, HIV particles or don't have HIV particles in your bloodstream. Here is a variation called a sandwich, which, is sort of a dangerous thing to say so close to lunch. But in this assay, you have the antibody absorbed to the well, you have the patient sample and you bind the antibody, and then you do a sandwich where the secondary is really another primary. Okay. Now you might be thinking, gosh, that seems complicated. Why would we do that? Well, this type of thing is actually commonly done in some of the over-the-counter ELISAs that you might be familiar with. Okay, so such as, oh shoot, I'm so sorry about that. Hopefully no one had a stroke watching my slides flip back and forth. My mouse got a little crazy. This is an example of an ELISA, a sandwich ELISA. Okay, so this is a simple P test to find out if you're pregnant. There's an antibody that binds the hormone HCG, which is produced during pregnancy at high levels. And so there's already an antibody against it. And then if your P contains HCG, it comes across this and it sticks to the antibody. And then there's a chemical or another substance in the stick that's brought over, and it's the same antibody, but now it's got this color attached to it. And so this is why you get the pregnancy test result. Now, other than pregnancy testing, there's tons of other ELISAs that now are marketed over the counter. Diagnostically, in the lab, they're used lots. In research, they're used a lot. But you can go to the store right now and buy some ELISAs. You wouldn't even know they were called ELISAs, but that's what they are. So what other types of testing can we do over the counter, like at home for whatever it might be? Usually they're P tests. Write that into the chat. So we've got ovulation tests, right? Yes. LH testing. LH levels will tell you if you're ovulating or not. And if you're trying to get pregnant, then you can monitor them. There's a particular window of a few days when they should peak, and then you know it's time to inseminate. Drug testing of various kinds. So THC can be detected. Opiates can be detected. Benzodiazepine can be detected. What's the other one? There's another one I don't remember right now. Cocaine can be detected. There's a bunch of them. Oh, and some people are mentioning they have a test for ketosis. Yes, you can test for ketosis as well. You can do that at home too if you're actually trying to induce ketosis in your body. So these are some examples of things you can test for. You can test for people who are, can test for amphetamine levels and things like that. And it's all based on antibodies. So as long as there's something that has a specific antigenic response, you can test for it. Okay, so we are going to take our break, and when we come back from our break, we're going to do a Kahoot. So when you come back from your break, please be ready for the Kahoot. I'm going to post the pin up here in just 1 second. We're just going to take an 8-minute break, so we'll come back at 11:15 to do the Kahoot, and then we'll do our class review. First question. Which of these is an example of natural or passive immunity? It was transplacental IgGs, so natural, right? Some of you picked this one, catching the flu, which would be natural, but that would be active because you have to make your own antibodies and memory cells. Which of these is found on the surface of T helper cells? T helpers. Which of these is found on the surface of T helper cells? T-cell receptors. MHC2 is found on antigen-presenting cells, not on the T helpers. It's what the T helpers look at. CD8 is found on cytotoxics. Okay, next, let's see. Parasitic infection is associated with an increase in which cell population? Eosinophils, that's right, eosinophils. Basophils are associated with allergic responses. Lymphocytes would be if the adaptive immune response had kicked in, and you might see this actually, but this is a definite giveaway. Red blood cells, of course, no. Sometimes they go down. A mutation, which of the following would cause the most severe loss of immune function? Good job, you guys. C3 it is, because remember that's the guy that's responsible for turning on the cascade that leads to all the activities and is also involved in all of the 3 different pathways that come in. Okay, this is a multi-select. Which of the following is required for T cytotoxic cell function? For them to do their job, what do you need? Like all that are true. So all of these are true except not this. Antibodies do not need to bind their antigens for T cytotoxics to do their job. Which of the following occurs during T-independent B cell activation? Is binding of an antibody to antigen, and remember that's really the only step, because unlike T-dependent B cell activation, we don't require a T helper cell interaction, which would this be, and we don't produce memory cells. Which of the following is not true about B cells? Which of these is not true? Good work. So B cells are specific. Each B cell has thousands of antibodies. The antibodies are all specific to one antigen. So they can only respond to one antigen each. Which of the following is part of the second line of defense? Is phagocytes, right? So skin and mucus, part of your primary or your first line of defense. B cells, part of your third line of defense. To grow a virus to create a vaccine for human flu, you could do which of the following? Is this one, right? So no viruses can be grown this way, period. This would only work if you were trying to grow a phage, which is not a human pathogen. Which of these is a step in a positive ELISA? So yes, all of these steps. So the first thing that happens is the antibody binds the antigen. That's the primary antibody. The second is the secondary binds the primary. Antibody binds antibody. And then that secondary antibody brings in the enzyme, and then you add the substrate and it cleaves or does not cleave it. Multi-select natural killer cells. What's true about them? Select the things that are true about natural killer cells. And pay no attention to my typo. Sorry about that. Hopefully doesn't change the meaning of the sentence too badly. All right, so yes, they do use the same tools, perforin and granzyme, as T cytotoxic cells. They're derived from the same lineage, but they're part of the innate immune system because they are not specific. They don't recognize antigens. What they do is target the pattern, cells that don't have MHC1. Multi-select, DTaP, contains heat-kill toxin and a protein isolated from pertussis. Which type or types of vaccine are in this shot? So yes, this heat kill toxin is called a toxoid, and this isolated protein is a subunit. It's not a whole agent, and it's not a whole agent. So it's not either one, attenuated or inactivated, which again isn't usually done for bacterial ones. What's the most effective type of vaccine in people usually? Remember, I'm saying in people. Nice work, guys. Whole agent attenuated it is. True or false? Whole agent inactivated carries a risk of back mutation to virulence. Well, this sentence is poorly constructed. Carries more of a risk. Whole agent inactivated vaccine carries more of a risk of back mutation than whole agent attenuated. True or false? Apologize for that. I wrote these last night. I don't know if you can tell. It is false, right? Whole agent inactivated has been heat killed and so can't back mutate. The attenuated one is the one that could back mutate. All right. Our bodies produce all of the following immune proteins except not which one? Yes, siderophores, that's produced by bacteria to bind iron. We produce transferrin. Okay. Interferon alpha and beta function to what? Interferon alpha and beta function to do what? Yes, they warn the neighbors. They're secreted by a virally infected cell to warn the neighbors. All right, last question. C1 is required for what? Nice work. It is the classical pathway. These other ones, lectin and alternative, don't use C1. All right. So let's see who gets bragging rights today. And of course, remember, I know that you were number 4 if you're not on this podium, so you should still feel proud of your accomplishments today. Good job, Kylie. Okay. Oh, it's showing runners up. That's new. All right, good work, guys. let's go back to our here. And so we are going to have a review and a Q&A, but before we do that, since this is the last time we're going to meet as a whole group, I just want to say that you guys have done a phenomenal job of working under pressure in very unusual circumstances. And I have had a really awesome time working with you. Probably I've had more fun than you have. Sorry about that. Some of the things that we're doing are just brand new. So when you go to fill out those evals, you know, I appreciate all constructive feedback, things that you liked a lot or things that you didn't like, or you were like, this would have been better if you had only done this thing. We're just working as hard as we can to kind of create a robust, rigorous, useful educational experience for you, despite the fact that we can't meet in person. So I want to thank you for all of your hard work, because I know that you have all had all kinds of things going on at the same time as this class. So I appreciate you showing up and putting in your good faith effort. And regardless of what the academic outcome is for you, I know that you tried. And you know, if you end up having to retake the class, then I'd be delighted to work with you again. But if not, then good luck in all your future endeavors. And don't be a stranger. Feel free to reach out to me just to say, hey, this is what's happening now in my life. I have people now who are graduated and just joined the nursing ranks this season to become part of the COVID frontline. And so it's really been exciting and nerve-wracking for me to watch them go through that. But I wish you all the best of luck and thank you so much. All right, so with that, let's see what you got for me in terms of review questions, and let's devote the next like 20 minutes to review for the test, unless we run out of things to talk about, and then we'll stay for a Q&A if you have questions about like the lab report or things like that. As you know, the test is proctored here in this time slot. If you have a conflict with that time slot, email me if you haven't already, and then we'll work something out. All right, so let's let the question period begin. What's on your mind? Raise your hand if you've got something you want me to talk about. All right, let's start with Eveline.
I had two questions regarding the pathogenesis section. The first question in the study guide was, what are siderophores and how do they promote diseases? Do they basically promote diseases because they have a stronger pull on the iron, therefore they're able to use the iron for their metabolic growth, and that's how they promote their disease because they can actively grow?
Yeah, and it's, yes, so they create the siderophores, or they have the siderophores so they can pull on iron, use it for their own metabolic growth, just as you said. And it does tend to be successful if they can find spare iron. A lot of those organisms also will deliberately break red blood cells to get more iron out.
And then for endotoxins and versus exotoxins, I just couldn't figure out which of those could be detected by antibodies and when are hosts exposed to them.
Oh, yes. Okay, so let's start with the difference between them at the chemical level. What is an exotoxin made of?
Protein.
Yes. An endotoxin is made of?
Lipid A.
Right. And so antibodies need something specific structurally that they can bind to, and lipids don't really have those kind of like really obvious unique structures typically, especially not fatty acid based ones. So proteins and complex carbohydrates are antigenic, but lipids generally are not. Okay. And so based on that, which would you say generates an antibody response?
The exotoxins.
Exactly. And now think about when your body would be exposed to them. So the exotoxins are proteins being made continuously in the bacterial cell and being secreted. So as long as the bacteria is alive and well in the body, it's making this toxin. But endotoxin is what?
It's only released when the cell dies.
Right. So that will happen, right? As the bacterial infection progresses, some of the cells get older and they die, but it's sort of gradual. Your exposure is gradual. If you were to have a mass killing event like with antibiotics, then there would be temporarily a point where a lot of endotoxin was released suddenly, but then it would go away, right? Your body would take care of it. So that's kind of the difference in terms of when you'd be exposed to it. Does that help?
Okay. And then for like which cells produce them, are there like a specific name of cells?
Oh, that's more just referring to, you know, again, the cells producing it from the DNA as they're alive versus it just being a part of their structure that is released when they are dead. Yeah.
Perfect. Thank you.
You're welcome. Haley, what's your question?
Hi, can you hear me?
I can.
So I was just wondering, when we went over the subunit, I couldn't quite get all of what was written down on the slide. So the subunit is safer, but it's less effective because...
Okay, so what is a subunit vaccine? How is that different from a whole agent?
I don't know yet.
You're not sure. Okay. What do you think based on the name, like whole agent, like if it was a virus was a whole agent, what would a subunit maybe be?
Not the whole thing?
Yeah, exactly. So a piece of it, right? Maybe one protein, a viral spike, or maybe, you know, if it was a bacterial situation, maybe like one protein from the bacterial membrane or one piece of its capsule, right? So the subunit is just a small piece of something. If we think about what is antigenic, a whole agent has all kinds of things. Even a virus, which is relatively simple and small, is going to have a whole bunch of antigens in the capsid because there's probably multiple proteins that make the capsid. And so they're going to be lumpy and bumpy bits that can be bound by antibodies. The spike is going to have a couple of different types of antibodies that can bind to it. So there might be like 10 antigens or more. And if it's a bacteria, there's going to be hundreds. But a subunit is only one usually, or maybe two. So based on that, if we think about what our body's actually responding to, if you give it a lot of options versus just one option, which one's going to be more effective? So does that help, Haley? Or
Yes, thank you.
yeah, okay. Great question. Genesis, what is on your mind?
I have a few questions.
Excellent.
Sorry.
That's what it's for today.
Okay, so for the complement pathways, those pathways that lead to, or that involve two types of C proteins, I remember you saying that, like, so it's not an all or nothing sort of event, I guess. So like you don't need both like for the inflammation, you need C3, one of them, and C5's one of them. Sorry. Do you understand what I'm saying? Sorry.
Not 100%, though I can jump in and comment on some things, but go ahead and
like a mutation where you couldn't make like one of those. So like C3, whatever is involved. You're saying, I think I remember you saying like you could still kind of proceed with inflammation because you still have that C5 available.
So are you kind of getting at like, how would you know what the effect of a particular C protein mutation would be?
Yeah, sort of.
Or which one would be worse, or?
Not which one is worse, I guess. So though, shoot, sorry. So like those pathway, so like inflammation involves it's not C3, it's C2. No, C.
Oh, so you're trying to remember which things do what?
Yeah.
Okay,
and
great.
Like,
yeah.
Those
so
that require two.
Yeah. So when we're talking about the different outcomes of complement activation, as long as all the C proteins are there, as soon as C3 becomes activated, then everything does happen because it's a cascade of events. But the cascade matters if you were to ask, well, what would happen if you didn't have C2, or what would happen if you didn't have C9? So that's why we want to know the details. But basically, C3 becomes activated by being split in half. One is an A, C3A, and one's C3B. And C3A is the inflammation one. And this is how you remember that, or at least how I remember it. So take your own mnemonic to use, but inflammation, C3A. is involved in inflammation. And then C3b is involved in opsonization.
Okay.
Now, also, C3 activates C5, which splits in half, and C5a is involved in inflammation, and C5b is involved in cytolysis. Now, if you're asking me about whether inflammation can proceed if you only have one of those, right? Because inflammation is triggered by both C3a and C5a. So do you need them both? No, you don't need them both. But if you don't have them both, then the inflammation level is less for two reasons. One, it's just an additive thing, right? Like C3a and C5a both bind to receptors on basophils that release histamine. So if you only have one of them, you're going to have less histamine, and histamine is an inflammatory agent. But also C5a, in addition to triggering histamine release and triggering inflammation, is a chemoattractant for phagocytes. So that makes it more likely that the phagocytes will be able to find the source of infection. So does that help?
Yeah, that's kind of what I was pointing at. Like, it's not a all or nothing thing. So if both of them aren't there, if one is still there, then it still can kind of
it can limp along. It's not as good, but it's still going to give you some inflammation, but you'll see reduced inflammation. And then, but for some things, like if you get rid of C5, you'll still have some inflammation, but you have zero cytolysis. Because even though C5 works with C6, 7, 8, and 9, without C5 to organize them, they just don't do anything.
Okay. And then also, can you describe or like explain the difference between focal and systemic infections? I guess I'm kind of like getting caught up.
Yeah. So the way you want to think about it is at the end, focal and systemic look the same because they're both systemic. But focal is something that started out local and normally would have stayed local. It's just unlucky. You know, like if you get a paper cut, that's usually local. If you skin your knee while hiking, that's usually local, right? If you have an ingrown hair, it's usually local. Those normally stay local. Normally the body can control the infection at the site and get rid of it. But sometimes if the cut is deep enough or the person's immune system is weakened or they just got unlucky or the dose was really high of the infectious bacteria or the particular species was very aggressive, sometimes that normally local infection goes systemic. In that case, we call it a focal infection. Does that help?
That does. So is the only way for something to like be systemic without taking that focal path is like if it just went directly into the blood or lymph? Yeah.
And some things normally do that, right? You know, some diseases are just, as a rule, they're just not local, right? Once you get the measles, you don't just get like the measles in one cell and it sticks around and then you're fine. Not usually. I mean, I suppose you might say, well, there's asymptomatic people and maybe that's why they're asymptomatic, and I can't argue with you. But typically when we diagnose someone with the measles, we're like, oh shoot, we're going to have a whole systemic situation here. Typically, I'm sort of blanking on an example, but a good example would be tetanus. Like tetanus doesn't stay local usually. If you get tetanus, yes, the bacteria are confined, but their toxin production goes everywhere. And that's just common, right? So it's not like you probably are fine. It's more like you're probably not fine. Does that make sense?
Yeah. And then do we need to know the mechanisms of how virulence factors work, or just like how they generally promote disease?
Just the level of detail that we covered, right? So like fimbriae are considered a virulence factor. What do? They attach to things. They help the bacteria bind to a tissue and they're not washed away. What does the M protein do? You know, what does the capsule do? It inhibits phagocytes. What does, so those kinds of things. The level of detail that we talked about in the lectures, all you need. Does that help?
Yes. And then, sorry, one last question. Yeah. The distinction between droplet and airborne.
Oh man. Yes. Well, it's a little murky, and basically, and it's a little bit outside my expertise. For people have fights about this, but basically droplets are bigger. And if it's considered airborne, it's a micro aerosols, and they go faster, further distances, is kind of my understanding of the difference. There might be more sophisticated, better definitions and comparisons that can be made by somebody in the field. But that's my understanding, is that although the terms are often used interchangeably, and in this class we didn't make a distinction and we're not going to, technically droplet transmission is in droplets that are larger, like the size of the droplet of the liquid that's floating around in the air is larger. So it tends to fall out of the air sooner.
Okay, so size matters, but in this class, we're not.
Size matters, but in this class, it doesn't. And if you ask a true epidemiologist, they might give you a better or different answer than that. So I don't want to nail myself to a tree on this one, but that's my not very sophisticated understanding. And it's a great question. I have often wanted to have a longer conversation with someone who's actually like in the field to say, hey, what are you all using as your definition these days?
Okay, thank you so much.
You're welcome. Okay, so we have Haley again. What's your question? Oh, I see your hand went down. Was that just an accident, Haley? Okay. If you do have a question, Haley, I'm so sorry I can't hear you, but I'm just assuming you took your hand down, so that's quite all right. Iveline, what's on your mind?
So I just had one question in the epidemiology and like a couple in the innate immunity.
Yeah.
For distinguishing between antigenic drift and antigenic shift and how it applies to the rapid pace of the influenza, is that basically because the influenza does antigenic shift where it changes its antigens continuously, and that's how it avoids being detected by the immune system, and that's how it's able to evolve?
Yes. So antigenic drift is slow and it happens in many organisms, including viruses and bacteria. And if it's an RNA-based virus, it happens a little bit faster because it's basically just the error rate during replication, creating new variations of the proteins, and it's slow, like one nucleotide at a time, meaning one amino acid maybe per generation. And antigenic shift is faster, and not every organism does that. That's not necessarily happening all the time, but in the flu it happens. And shift is kind of not just one mechanism, but what it means is that it's a faster mechanism. It's not just error replication rates, like you make a mistake during copying of the nucleic acid. In the flu, we looked in more detail about how exactly that shift is happening, which is to say, because there's multiple pieces of the genome and they are multiple hosts, they can mix and match their pieces, and you can have like an entire protein changed in one generation. And so shift is not happening all the time in every organism, just some, depending on their details of reproduction. And it is much faster. Now there's other mechanisms of shift we didn't talk about. So there's some eukaryotic parasites that do antigenic shift, and they do it differently. They actually have a mechanism that's kind of like the B cell mechanism, where they like deliberately mutate and shuffle the DNA in their protein coding regions. And so that's something that's not common either. But the parasites that do that are kind of a pain to deal with. Does that answer your question? And did I miss part of your question? And I'm sorry if I did.
No, you answered it perfectly. Thank you.
And did you have a second question as well?
Yeah.
Okay.
So just to make sure I'm on the right track, describe inflammation, its hallmarks, and the role of vasodilation. So is the role of vasodilation basically it makes more room for the blood vessels, so then more blood and then more cells can come to the scene and handle it?
Yeah, vasodilation means that the blood vessel enlarges, and that causes the spaces between the endothelial cells to be larger. So it makes it easier for white blood cells to exit and for fluid to exit the blood vessel, yeah.
So is it the white blood cells that come to the scene along with the red blood cells?
No, red blood cells don't have the same abilities to leave, and you don't want them to. That's basically hemorrhaging, and that's not good news for you. So the process of diapedesis that we talked about, where the white blood cells squeeze out, is like they get themselves really, really, really skinny, like, and then squeeze out. That's not something red blood cells do.
Okay.
But the fluids, yes, the fluids, you do get fluids leaving. Yeah.
So the fluids leaving the blood vessels going to the site of inflammation.
Yeah.
Okay. And then for fever, I know that the advantages are like increasing transference, increasing the IL-1 activity, and producing interferons. But I'm not sure what does IL-1 do.
Oh yeah, and we didn't talk about that. So basically, and you won't be tested on this, but white blood cells talk to each other with chemical signals. So instead of like picking up the phone and talking to each other, they send a chemical signal out that's a message to other white blood cells. And that's what interleukins are. Interleukins are a special type of chemical, interleukin, between white blood cells. So there's a bunch of them. And depending on the type of chemical it's sent and depending on who receives the signal, different cells can respond to them. And interleukin 1 has a bunch of effects. It talks, though, it's part of the loop of talking to your hypothalamus, that is the part of your brain that controls your thermostat, like your body temperature. And so it's part of that whole process of talking to your hypothalamus and resetting your body temperature.
So then if you have a fever and it produces interferons, so does those interferons tell the neighboring cell that, oh, we have a fever, or like?
So the production of interferons in this case is not alpha and beta. The interferon is gamma, and it causes macrophages to be more active.
And then, so it says what is the role of lysosomes in the innate immunology? Does lysosomes destroy the peptoglycans in the bacteria?
Yes, and let's make sure we're using the right term, lysozyme. Lysosomes is the organelle. I know that's what you meant. And unfortunately, it's another one of those terrible words in biology that's the same and or sounds very similar. Ah, what a pain. But lysozyme is the enzyme, and it does exactly what you said. It targets peptidoglycan, and it breaks the bond between the two sugars of the filament. So it breaks down the cell wall of bacteria.
Okay, thank you.
You're welcome. Great questions. Okay, next we have Simple Ann. What's your question?
For the lab report, is there a specific format? Like, okay, so part 1 is introduction, method, conclusion, or procedure?
Yeah, great question. And I'm just going to table it for 1 second so I can sort of clean up any last questions about the review, but I do want to talk about that. I think lots of people are going to want to hear the answer to that, but I'm probably going to have you restate it because I don't multitask very well. So let me just ask a final call for any last questions about the material, and then let's move on to the Q&A about labs and other assorted things. So I'm just going to check in with Kylie to see if you had a question about the course material, and then we'll see.
Yeah, it's probably gonna be a long one because I'm still confused on the transformation efficiency equation.
Oh, thank you for bringing that up, and I forgot to announce this, but I will. I'm not putting that on the test.
Oh, yay!
Because my intention was to talk about it in class, and I didn't have time. I mean, I could have done that today, but I decided it was more useful to table that and instead do a review. So the only place you need it for is for the lab. So I'm happy to walk through that for full understanding of the lab. But again, let's put that on aside and just see if there's other questions for the review for the test. And thank you so much for mentioning that. And I need to make myself a note because I forgot to send that announcement out, and you just reminded me of that. So I'm just gonna make sure I do that. There's no TE.
Definitely makes it easier.
Okay, did you have any other questions?
Nope, that was the only one.
Okay, so I'm happy to address that, but let's just check in with Serena and see if she has a question about the course material.
I had a question about kind of what you thought Genesis was talking about before with how do you know what mutation will affect, or what C3 or C2, or if you don't have that, like how do you know what that will affect and what kind of mutation it will be?
Yeah.
Does that make sense?
So you want to make sure you have that little diagram of the complement and you know which proteins are involved in which pathways. C3 is like absolutely required for everything, but there's different ways to activate C3. So if you're going upstream of C3 to C1, C2, C4, C1's only involved in the classical pathway, which means that without C1, you're going to lose that form of activation. That means if an antibody binds antigen, you won't be able to initiate complement. But the other pathways are still intact, so you can still get all three outcomes of complement if it's a bacterial pattern that you're responding to instead. For C2 and C4, they're going to affect the classical and lectin pathway, but they won't affect the alternative pathway. So that still leaves the possibility of complement to be activated if you're responding to a polysaccharide lipid signature on a bacterium. And BPD is involved in the alternative pathway. So, it's sort of like the different pieces tell you whether or not the complement can be activated or not. And so for the ones that are upstream of C3, it's a matter of, can you activate complement under what circumstances? Downstream of C3, you're going to have more specific effects where you're not affecting how to turn on C3, but what you're affecting is what happens when you turn on C3. So if you lose C3, of course, nothing happens anywhere. There's no complement at all. But if you lose, let's say, C5, you're only going to lose cytolysis and half of inflammation. And if you lose like C7, you're only going to lose cytolysis. So it basically comes down to knowing that diagram really well and being able to say, okay, well, without this, what's the role of this thing? Is it involved in everything, half the things? Does that help?
Yes, because I had that list, but I was like, I don't know. But now, like looking at like the diagrams too, because I actually drew all of those pictures out for classical, alternative, and lectin pathways. So I have everything numbered and labeled.
Yeah, and that's exactly what you need. You need that diagram, and that's the most helpful thing.
Okay, thank you.
Awesome. Eveline, did you have a question about the course material?
Yeah, I had a couple more questions on the adaptive immunology, if that's okay.
Yeah, absolutely.
So the first one, comparing T cells and B cells. So do B cells use antibodies to attach their antigens, and do T cells use T cell receptors to bind to the antigen? So the T cell receptor helps the T cell bind to the antigen, and then the CD helps them to look inside the basket.
Yeah, so the way that it works is the T cell receptor binds the antigen, but it can only do so inside of an MHC. But it can't find the MHC on its own. So the job of the CD is to find the MHC so that the T cell receptor can look inside of it.
So the CD helps the T cell to find the MHC basket so that the T cell receptor can look inside to it and bind to the antigen.
Exactly.
And then it says here, what's hive free versus endogenous? Can you explain that?
One second, hold on. There's just a situation in my house and I will be back in 1 second. All right, I'm so sorry. Can you say that again?
Yes. In the study guide, one of the questions was saying compare contrast T cells and B cells and what type, free versus endogenous.
Oh, yes. So which one? Yeah. So this is about whether basically B cells can bind antigens that are floating by and T cells can't, because T cells have to have the antigen handed.
Okay. And then comparing and contrast like the T independent versus T dependent and the role of T helper, MHC2 antibodies, proliferation, differentiation, plasma, and memory cells. Is that basically where like T independents can go ahead and go bind to the antibody without verifying, without the help of the T, without going to the T cells to ask for a second opinion just because they're super overwhelmed. And then with T dependent, they need the second opinion of the T helper. And then when the T helper says okay, then they can like proliferate, and some will become plasma to make the antibodies, and some will become memory.
Yes. And the important difference is not only that the T independent doesn't require the T helper in order to make antibodies, but also because it's not talking to the T helper, it doesn't get the signal to proliferate and to make memory. So instead, it's just this one guy kind of going, antibodies! and then forgets about it.
Okay. Okay, yeah, and then T cells identify their specific antigen targets with the T cell receptors.
That's right.
Cool. Thank you.
You're welcome. Okay, let's check in with Justice.
I have a question about the unknown ID lab, so it can wait.
Okay, we're almost there anyway. Genesis, what's your question?
Follow up with the, sorry, blanking right now. The CD protein, CD, yeah, still working on it. So do they actually like bind the MHC, or I kind of thought of them as a signal, but in your Kahoot, I think you had them binding, so I was confused on that.
It's more sophisticated than I'm letting on, and just to keep it simple, we're just going to say that they bind. So the CD binds the MHC, and TCR then can bind the antigen.
Okay, thank you.
That's how we're going to do it, because we don't need to get into the murky details unless you want to be in that field, which is a super fun field. Go ahead. But let's just keep it simple here. Yeah. Okay, let's see. So I want to go back to, we're going to now, right on the nose at noon, we're going to switch over to other questions. Feel free, though, if you have a question about course material, you can always ask that. But all those other lab-related stuff or lab report stuff that's on your mind, the unknown ID project, whatever it might be, we're going to kind of do now.